A short talk by Kingini Roychowdhury. It's going to tell us about underlying principles of chromosome organization.
Good evening. My name is Kinkini and I'll be talking about entropic demixing and organization of tomopolymers, or again topologically modified polymers, by the formation of loops. And we'll find out whether we can have some physical principles that can explain the underlying some of the features of chromosome organization. So as mentioned by Professor Apratim a few talks back, that two ring polymers present in a cylinder segregate to two halves of the cylinder, and this has been recognized as one of the primary mechanisms of daughter chromosome segregation in E. coli bacteria. But there are also spherical cells like the S. aureus, which also needs to perform chromosome segregation. So my question is whether topological modifications to the ring architecture can help this sort of segregation and help this. So I'm not talking about a particular system in this talk. I'll rather be focusing on the simple principles where we can change topological modifications to tune entropic interactions, and then find out whether we can explain some features of chromosome organization using this. So it's answering simple questions like whether the changing the size of the loops and the arrangement of the loops matters. So we mainly work with bead spring polymers, and we only have bond interactions and self-avoiding interactions in the simulation so that we have no active force in the system. And we deal with three kinds of topologically modified polymers. One are these asymmetrically arranged small and big loops, where the small loops are present on one side and the big loops are present on another side. Second are these symmetrically arranged loops, which we have nicknamed as flowers. And the third are these loops arranged on a linear chain. And we'll find out the effect that each of these architectures has. Now, we look at demixing. For this, we focus on two polymer systems. In one system, we have two ring polymers. In one system, we have two of these flower architecture. Both of them have the equal number of monomers and same volume fraction. We see that for ring polymers, they are overlapping and they interpenetrate into each other. So there is no demixing present for simple ring polymers in this system. However, if we look at this architecture, we see that although the two polymers are moving around, because of the presence of this architecture, they do not interpenetrate or overlap, and they remain relatively demixed. So we see that architecture can enhance the entropic demixing of polymers. So to quantify it and to understand how different architectures can cause this, we have first plotted the monomer distribution of one polymer around its own center of mass. So it's a two polymer system. We've taken the center of mass for one polymer and we plotted the distribution of its own monomers. So near the center of mass, obviously there is higher density and then it drops. But what is interesting is that for linear and ring and even this random linear loop, we see that the density is much lower at smaller distances. And ARC16, which has the same number of loops as this, but just that they are well designed and they are clustered at one end, the density near the center is much higher. So just the creation of this architecture is causing compaction of the monomer around its own center of mass. So even though they both have the same number of loops, this architecture is better at compacting the monomer. But does that mean that it can separate that polymer around the other monomer? For that, we have plotted the distribution of the other monomers of the polymer around this center of mass. So this gives a measure of how interpenetrated the two polymers are. If there is a high density of monomers of this polymer near the center of mass, that means that that polymer is more interpenetrated into the other polymer. Again, we see that at small distances for linear ring and this random linear polymer, there is a higher density near the center of mass of the other polymer. And for ARC-16, there is very low density, so there is very less interpenetration. Then the density increases only after a particular distance from the other polymer's center of mass. So we can say that our architecture creates, or the clustered loops creates, increases the degree of demixing. So to analyze contacts between these polymers, the usual way are contact maps, but to compare between different architectures, it's very difficult. So instead, we do a different analysis where we take the number of total interpolymer contacts. Suppose one monomer from this polymer, one monomer from the other polymer are within a particular distance, that is one contact, and we count how many such pairs are present at a particular time step. Then we plot the number of times the total interpolymer contacts are between 0 to 100, number of times it between 100 and 200, and so on. To simplify this, we have taken a threshold of 150, where 0 to 150 is low contact and greater than 150 total interpolymer contacts is higher contact. We see that for ARC-16, this and flower, we see that the fraction of time the polymers are in low contacts is much higher. So they usually remain in very low contact with each other, whereas linear ring and this architecture are mostly in high contact with each other. So this establishes how the creation of these well-designed loops can cause repulsion in between the loops and can influence its segregation. But does it also somehow ensure radial organization of particular segments inside the sphere? So for that, we have taken an architecture like this, and we have looked at the preferential localization of these loops from the center to the periphery of the sphere. And the red line plots the density of the big loop, and the small and the blue line, the red line plots the density of the small loops, and the blue line plots the density of the big loops. And we can see that when there is only one polymer of such kind in the system, the small loops have a higher density near the center, while the big loop is spread out near the periphery. But if you take multiple polymers in a system, which is again inspired by multiple chromosomes in a eukaryotic nucleus, you see that the small loops avoid each other and face the periphery, while the big loops are overlapped near the center. So going from a one polymer system to a multiple polymer system changes the radial organization patterns of the different loops. But if we change the architecture, suppose we go from a 3-loop architecture like this to a 6-loop architecture, this is a more compacted form of this. And we see that compacting the small loops further increases the degree of radial localization of the loops, where the small loops are found even more probabilistically near the periphery. So more compact loops equals stronger radial localization. And if we plot the total monomer densities, small loop, big loop combined as a function of radial distance, we see that there is a slight increase of the total density near where the smaller loops are located. We believe that this is reminiscent of heterochromatin-euchromatin localization. However, the difference is very slight. But as we move closer to a more accurate biological system with hierarchical loops and other processes present, this increase might be enhanced. Finally, a nucleus is not always spherical. Mechanical stresses can often deform nuclei and cause them to get flattened. Experiments have shown that a more flattened nuclei can lead to differential gene expression, and that can in turn stem from changing contacts in between the chromosomes in the system. So to that end, we have also studied ellipsoidal systems, and we have analyzed contacts in between different pairs. Now, I'm not going to detail into this data, but if anyone is interested, I can explain later. Finally, I'd like to thank my colleagues, especially Snigdha and Shirang, and my mentor, Dr. Apratim, for their support and contributions. And thank you to the organizers for giving me this opportunity. Thank you.
Thank you very much. Are there questions? Anjana, please.
That was really nice. Can you talk about how you think these various arrangements form? And is it possible that in a biological system, there are transitions between a flower structure and a 3-loop structure and a 1-loop structure?
Yeah, so there are these loop extruding proteins and SMC proteins that forms loops and increases them. Then on top of that, within a loop, there might be other loop extruders present, which causes hierarchy. So this 3-loop structure can get compacted into a 6-loop structure. So in biology, those loop extruding proteins are found in both eukaryotes and prokaryotes. So that's how I would assume that the structures change.
And in case of your simulations, have you considered a scenario where there is actually dynamic
No, we
transitions
haven't.
between these loop structures?
Yeah, so we haven't looked at changing systems yet. Till now, we have definite structures to understand the underlying principles first. And then we plan to introduce more dynamic loops in this so that we can model that effect as well.
Okay, thank you very much for a very nice presentation and congratulations on your paper.
Thank you.
All right, thank you.